You had everything in your notes.
Your part was difficult.
And why? Ntabwo mwari mwabyize se? Sinari nabibahaye rwose.
It was somehow complicated.
Okay. Cyangwa mwariraye mwanga kwiga?
No.
Twarize mu buryo bushoboka ariko yabana akagozi karacika biranga.
Kandi nababwira ibyo mwiga kweli.
Hano icupa ritaba dukenewe.
Uducupa twa mudo. Tumarisha iki? Nzabishyira kuri mudo past papers zizajya.
Kabisa.
Just. You are right to go kubona past papers. Mufite ubwoko ko muzatsindwa se mugasubiramo ko mutangiye gusaba. Izi ryo ntazo mfite. Nzabaha ikizarangira. Okay, so let's start. Okay, so the course of today is clinical chemistry 1. So am I the first one, or you already started with the Evelyn?
You are the first one.
I'm the first one, so I'm going to. Okay, this course is going to cover different chapters. Okay, this course is going to cover different chapters. So among the chapters that we are going to see in this course, the chapter 1 is going to be the hormones. So I'm going to teach you about the hormones and the enzymes. So Evelyn will teach you the rest of the course. So this is the clinical chemistry 1. So is it your first time to hear the term hormone?
No.
You studied it in secondary school?
Yes.
Okay, then it's going to be easy. If it is not first time. In glands. So endocrine glands, so the law of this is homeostasis is maintained by both nervous system and endocrine gland in human body. So hormone, there are the substances that secreted by these glands, of course, and some hormones are regulated in nature, like tropin hormone, releasing hormones. So there are two systems that which coordinate the communication through the body. Those systems are endocrine glands and the nervous system. So the endocrine gland, you know that they secrete hormone, and nervous system, they convert high-speed electrical signal along with special cells called neuron. So the nervous system it control rapid onset in limited period of limited time, in short time, where endocrine gland slow onsets, but it take time. So this is an example. So the endocrine signaling is just one of several ways of information to transmit between animal cells. You know, I taught you about the signal in cell signaling. So endocrine is also one of the cell signaling. You remember we studied about the synapses, gap junction, those signaling we studied in biochemistry. So this is also going to be another cell signaling, how the hormone are transmitted from one cell to another one, or from the gland to the cells. So intercellular communication, the way signaling are transmitted between animal cells and classified into criteria. So one is type of secret cell and the route taken the signal reaching the target. So endocrine signaling, so there is hormone secreted into extracellular fluid by endocrine cells, reach the target cell via bloodstream. So the hormone are released by the specific cell, and they are released into extracellular fluid. And then they reach target cells via bloodstream. So the main role of this endocrine signaling is to maintain homeostasis and mediate response to stimulus and regulate growth and development. Of course, you see different types of hormone. So this is the image on how these endocrine glands, so how the hormone are released into bloodstream and then reach the target cell. So this is endocrine signaling. There is also paracrine signaling, where the hormone, the signals of the substances are released by the secretory cell into the extracellular matrix, and then they reach the cell via the receptors. So this one. There is also autocrine signaling, where a cell releases the substances, which may be the hormone or and then these substances will again back and bind to the same cell, and then enter through the receptor. There is also synoptic signaling. So you know the synoptic signaling. So I think I don't know how, I don't have to talk about it.
So Alex,
yes.
hari abantu bari hanze nyine bakeneye kwinjira.
Icyo babatinze ariko.
Ariko ni ikibazo.
Okay. Okay, so there are many. There is endocrine, paracrine, autocrine, synaptic signaling, and neuroendocrine signaling. So where the hormone will be released to the bloodstream, and then from the neuron, from neuron, and then the neurotransmitters will be released to the bloodstream, and then from the bloodstream to the target cells. So this is also neuroendocrine signaling. So then let's see it one by one. Paracrine and endocrine signaling. So local regulator are molecules that act over a short distance, reach target cell swallowed by diffusion. So in paracrine signaling, target cell lies near the secreting cell. So endocrine signaling, target cell is also secreting cell. For example, endocrine signaling. Here is the target, the releasing the excretory cell. It will release these substances, the hormone, then they will go through the blood stream, and then we leave the bloodstream through the target cells. So the target cells are lying near the vessels. And is where the releasing cell or excretory cell will release the hormone, and they will immediately bind to the receptors without passing to the bloodstream. Endocrine signaling is where the substance will be released by the cell and then again enter the same cell. So synoptic signaling neuron from the specialized junction with eight cell called synapses. So the synapses translate molecular card neurotransmitters that diffuse. So for example here, this synaptic signaling, the neuron will release the neurotransmitters. The neurotransmitters will enter the target cell through synapses, the synapses. And also in neuroendocrine signaling, it's where the neuron will release the neurotransmitters to the bloodstream, and then from bloodstream to the cell. You understand? You get something?
Yes.
Okay, good. So there are endocrine tissue and organs. In some tissue, endocrine cells are grouped together in ductless organ called endocrine glands. So endocrine glands secrete hormone directly into surrounding fluid. These contracts with exocrine gland, which have a duct and will secrete substances into blood surface into the cavity. So those are endocrine glands. So hypothalamus, pituitary gland, thyroid gland, pancreas, ovaries, testes, and there are also other organs which may release the hormone. For example, thymus, liver, more. So last there are three major classes. The location of. So one will pass easily through diffusion, another one will need something to the transporter. So they are soluble, lipid soluble, and water soluble hormone. For example, the water soluble hormone, which are hydrophilic, there are these polypeptide hormone like insulin, and the lipid soluble hormone are the steroid hormone such as cortisol. So hormone classification. So as you can see here, there are steroid hormone, amine hormone, and peptides. So among the steroid hormone, we can see progesterone, testosterone, and estrogen. And the amine derivative hormone, here we see thyroxine, we see adrenaline, and the peptide is insulin. This is an example because we have many hormones. So then you have a cellular response pathway. So the water and lipid soluble hormone differ in the path of through the body. So the difference is how they pass and how they diffuse through the cells of our body. For example, water-soluble hormone are secreted by exocytosis, travel freely in the bloodstream, and bind to cell surface receptor. So water-soluble hormone will need cell surface receptor to enter the cell, where the lipid-soluble hormone diffuse directly close cell membrane and travel in the bloodstream bound transporter protein and diffuse through the membrane of target cells. So it is easily to diffuse through the membrane of target cells without being attached to the receptor. Okay, for example, here we have secretory cells, and this is the water-soluble hormone. Will go immediately through bloodstream, will travel through the bloodstream, and then it will bind to the receptor to enter the target cell. Once it enters the target cell, it will regulate the gene expression to give the response or to transcribe to something, which may be something we don't know according to the hormone that is entered to the target cell. So once it's entered the nucleus, it will regulate the transcription of gene and then give the response to the cytoplasm. This is water-soluble hormone. So about lipid-soluble hormone, this hormone will be released by the secretory cells, enter the bloodstream, bind to the transporter protein, and then leave the bloodstream, diffuse immediately to the target cell without binding to the receptor. Once it reaches inside the cell, it will bind to the signal receptor inside the cell, not outside the cell. So see here, this bound to membrane receptor, but this one enter immediately without binding to the receptor, and it's bound to the receptor, which is located internal the cell, inside the cell. So once, after binding to the receptor, it will enter the nucleus and regulate the gene expression, which will give the response to the cytoplasm. Okay.
Why the soluble hormone?
Sir?
Why the soluble hormone?
So they have a different characteristics. So the hormone have a different characteristics, as I said. Some may be soluble, another one may be not soluble. Maybe soluble to lipids, and another one soluble to water. So these depend on their characters.
Okay.
Binding of hormone to its receptor initiates signal transduction pathway leading to the response in the cytoplasm. So the enzyme activation and a change in gene expression, as I was saying, once the hormone binds to the receptor and then to initiate the signal transduction pathway, this will lead into the receptor in the cytoplasm and activate enzyme or change in gene expression. I think we studied a bit about the signal transduction. Don't you remember about signal transduction? We studied it in biochemistry. How an activator bind to the receptor on the cell and then activate the signal transduction, which will activate the internal pathway and the enzyme and then change the gene expression. I think we studied about it in biochemistry last time. So the same thing here, the hormone will bind to the receptor and initiate the pathway, so the signal pathways inside the cell, which will lead to the response in the cytoplasm, and then activate enzyme activation and change in gene expression. So I'll give you an example of signal transduction. And go to the nucleus. Okay. For example, here, this hormone will bind to the receptor, and the receptor will activate internal enzyme, which may be genus kinase, as I was saying. Once this enzyme are activated, then they will go to the nucleus and regulate the gene expression, which will result the transcription of messenger RNA to make new protein. But this will start by the binding of the cytokine on the membrane of the cell, signal transduction, gene transcription, and the release and the protein, and the release of messenger RNA for translation and making protein. Okay. So, for example, let's get to part 5 pathway. See here. This is cytokine, which will bind to the receptor. Once this receptor is conjugated by something from outside, and then it will phosphorate the genus kinase, the JAK2, and it will activate this STAT enzyme. The STAT enzyme will also be phosphorated to go to the nucleus for the transcription. You understand? Do you understand?
No.
You don't understand? But I think I told you about this. Ndababwira icyo bita signal transduction kugira ngo yumve. Hagomba kubaho ikintu ikintu dushaka, hormone which is released by secretory cells. A cell releases hormone, and this hormone will come ikaza ikabindinga kuri target cell. Target cell bifite receptor ariyo iyi ngiyi. Hormone yamara kubindingaho hano, immediately inside the cell signal ihitijamo. Iyo signal ije, donc byararitinga cell that something is binding to the receptor. And then the cell signal zimwe na zimwe zihita zihinduka. For example, I was giving you an example of this genus kinase. So it's a kind of enzyme which is located inside the cell. The enzyme hitiba phosphorated. Donc phosphor group hitiza ikabindinga kuri this enzyme. Imaze kuba phosphorated, and then nayo ihita itanga message ku kindi kintu gishobora kuba STAT5. Mfite examples nyinshi za signal transduction. Nyinshi hari. This I'm giving you an example of JAK2 STAT5. Iyamara kuba phosphorated, and then igatanga message here izahita ijya muri nucleus ikagenda igahita ikora ikaregulatinga transcription on the DNA, and DNA igatanga results messenger RNA, and then something ikaba yakorwa bijyanye n'ikiri targeted. This is one example. Another example is this one. Wicket PITK is another one. See, it's the same. There will be activation of the membrane by binding cytokine to the receptor, and then the AKT will be activated. PI3K will be activated to the AKT, REP, mTOR, ikamanuka gutya kugeza igihe izakora icyo dushaka. So this is actually this is signal transcription, transduction, and niba mutarabyumvise muri biochemistry, it's a bit complicated for you to understand ntago ari level yanyu, but I wanted to show you how it is done. Sibyo, you don't need to go into details on this transduction, signal transduction. Nagira ngo mbereke on how hormone will come and bind to the cytoplasm and then activate something inside the cell cytoplasm, and then result to the transcription. No murabyumva? Ntabwo mukeneye kubyiga cyane ibi ntabwo ari level yanyu, ariko hano nagira ngo mbereke how hormone nayo ikora ibi ngibi. So twari tugeze kuri ino slide. So I was saying that the binding of a hormone on its receptor, as I was showing you, initiates signal transduction pathway, leading to the response in the cytoplasm. The enzyme activation or change in gene expression. I think I showed you the examples. So the hormone like epinephrine has multiple effects in mediating body's response to short-term stress. So epinephrine binding to receptor on the plasma membrane of the liver, and then trigger release of a messenger molecule that activate enzyme and results in the release of glucose into the bloodstream. So this is an example of what we are saying, how the hormone will be released, how it will be transported through the epinephrine binding, how it will bind to the plasma membrane, and then they release the messenger molecule that activate enzyme inside the target cells, and it results in glucose into bloodstream. Okay? So here, this is the hormone, epinephrine, secreted by excretory cells. It will come and bind to this receptor of target cells. This will guanosine triphosphate, adenine triphosphate, and then secrete adenosine monophosphate, which will give results to giving to the release of protein kinase A and inhibition of glycogen synthesis or promotion of glycogen breakdown. I think you know the glycogen. So the role of this epinephrine is to increase the glucose in bloodstream. Yes? So this is the way. It is started by binding on the receptor, and then they activate this transduction, and up by inhibiting glycogen synthesis or promoting glucogen breakdown. So you know the glucogen synthesis. What is glucogen synthesis? Someone can tell us what is glucogen synthesis? So in case the energy is not used, it will be stored as glucogen. If the glucose is not used by our body, it will be stored as glucogen. In case the body needs glucose, then the stored glucogen in the liver will be broken down to give glucose. The complex then act as transcription factor in the nucleus, regulating the transcription of the specific gene. So the hormone will enter once to be regulated by the transcription. Okay. This figure also is going lipid soluble hormone will result by the transcripting something. So example of the hormone. So this hormone estradiol will come and enter as you have seen. This lipid soluble hormone doesn't need the receptor on a membrane. It enter through the fission. Once it enters, it will bind to the receptor, and the hormone receptor complex, it will go to the nucleus and start the transcription. By transcripting, messenger RNA will be released. For example, if it was the target, it was there for transcripting a protein like heterogenein, and then it will be translated as heterogenein. So heterogenein is a process of egg yolk that transport protein and some lipid from liver through the blood growing oocyst. There are multiple effects of hormone. Some hormone may have different effect on target cells that have different receptors of the hormone, different signaling transduction of the pathway. For example, here, epinephrine, as I told you, it will come to the target cells and break down the glucogen or stop the glucogen deposit. So the final effect is to release the glucose from the cell. So this is in the liver. Of course, the glucogen, when you hear a term glucogen, then think about the liver. Epinephrine on receptor, vasodilatation, and skeletal muscle, epinephrine, receptor, vessel, and the same thing, the same way epinephrine is transcripted to give something like, as I was saying, like a release of glucose, a breakdown of glucogen, like that. So the feedback regulation and the antagonist hormone pair are common in an endocrine system. So hormones are assembled into regulatory pathways. Simple hormone pathways. Hormones are released from an endocrine cells, travel through bloodstream, interact with specific receptor with the target cells, cause a physiological response. Even I even explain more. For example, a release of acid content of stomach into the duodenum stimulate endocrine cells to secrete secretin. This causes target cells in the pancreas and the gland behind the stomach to release pH. This is negative feedback. For example, I give you an example here. This stimulus, let's suppose in the duodenum there is a low pH. This will stimulate the S cells of duodenum to secrete hormone. So this stimulus will come to the endocrine cells, which may be S cells. The hormone will be secreted and will reach the blood flow to the target cells and it will give the response. So for example, once there is low oxygen, which is a stimulus in duodenum, the S cell of duodenum will secrete hormone called secretin. And the secretin will act on the pancreas, and then the pancreas will low or will release bicarbonate to low the pH, to increase the pH. This is negative feedback, and then in the simple neuroendocrine pathway, the stimulus receives sensory neuron, which stimulated the neurosensory cells. The neurosensory secretory cells secrete a neurohormone, which enter the bloodstream and travel to the target cells. Okay, let's also use the figure. This positive feedback stimulus, sensory neuron will act on hypothalamus or post-pituitary gland, which will release neurosecretors, which will act on neurosecretory cells to release neurohormone. This neurohormone will reach bloodstream to the target cells and give the response. Example, saccharin. For example, it will immediately activate the hypothalamus. Posterior pituitary secreted neurohormone, which is oxytocin. This oxytocin will come and act on the smooth mass of the breast and will release the milk. So the negative feedback inhibits response by reducing the initiate stimulus and preventing excessive pathway activity, while the positive feedback reinforces a stimulus to produce an even greater response. So do you understand what is negative feedback and what is positive feedback?
Yes, lecture.
Do you get something? What?
Yes, it's okay.
It's okay. A negative feedback, no kuvuga ngo hormone iba stimulated, a gland iba stimulated, ikareleasinga hormone, ariko ya hormone ikazaza nyuma ikongera ikaactinga kuri ya gland, ikayipreventinga nanone kureleasinga ya hormone. I give an example of thyroid stimulating hormone. Thyroid, the thyroid, okay, let's use this example. For example, prototropic releasing hormone secreted by stimulus. Hypothalamus stimulates the release of thyroid stimulating hormone by the entire pituitary gland, which in turn causes the thyroid gland to release thyroxine and thiodotyroxine, which is T3 and T4. T3 and T4, once released, cause negative feedback on the secretor of a thyroid stimulating hormone. You understand? So this hypothalamus, it will act on anterior pituitary to release thyroid stimulating hormone. Thyroid stimulating hormone is the one to cause the synthesis or the release of T3 and T4. So once these T3 and T4 are released, or once they are increased, they will again go to pituitary gland to inhibit the release of thyroid stimulating hormone. So remember, first pituitary gland cause thyroid stimulating hormone, and then thyroid stimulating hormone itumye habaho stimulation or the release of the T3 and T4. Ariko T3 and T4 in normal, they regulate many things like temperature, like body weight, anything. But iyo zibaye nyinshi zisubira inyuma zikajya kuri ya thyroid stimulating hormone yatumye zibaho zikainhibiting. That is negative feedback. For example, here you can see here there is a hypothalamus, anterior pituitary gland. There will be release of thyroid stimulating hormone. Once the thyroid stimulating hormone is released, it will act on thyroid gland, this thyroid gland, to stimulate or to release T3 and T4. So the increase of T3 and T4 concentration in the blood, once this is increased, it will inhibit the secretion of TSH, thyroid stimulating hormone. This is a negative feedback. And then positive feedback, I also give you an example. A head of fetus push against cervix. And then after, the nerve impulse from the cervix transmit to the brain. Then umwana amaze pushing a cervix, nerve, and the impulse, the message will go to immediately to the brain. And the brain will also stimulate